And this is some of the words that he mentioned during this meeting. The surgical side of transplantation of organ is now completed, as we are now able to perform transplantation of organ with perfect ease, with excellent result from an anatomical standpoint. All our effort must now be directed toward the biological method, which will prevent reaction of the body against foreign tissue. Later on in the field and in the early 20th century, this was actually a picture of the first kidney transplant. And this is the first human kidney transplant. This was performed in Ukraine and in 1933 by a prof named Vorony. The blood group of the donor was B, the blood group of the patient was O, so without even thinking there will be hyperacute rejection once you add this kidney. The kidney was obtained from a deceased donor, transplanted into the thigh vessels under local anesthesia. The warm time of the kidney was about 6 hours, but the kidney didn't work. Of course, it's not going to work. And the patient died 2 days later. Later, this technique became improved in 1952 by a French group. And instead of using the groin to transplant the kidney, they thought of transplanting the kidney inside the iliac fossa and using the iliac vessel for the anastomosis. And this French group has tested this technique into a son who was 15 years old. He was a carpenter and he has a single kidney. And unfortunately, he fell from the ladder while he was working and he got ruptured kidney. So his mother was his first donor. And they tried it to transplant the kidney. The kidney works for the first 3 weeks without any immunosuppression, but it ended by rejection and followed by the death of the recipient. And this is the picture of the first living donor. His name was Ronald Lee, and he donated his kidney to his identical monozygotic twin brother in 1954. This surgery happened in Boston, in US, and it happened 2 days before Christmas in 1954. This US surgeon was very proud of this surgery because they have seen that the kidney can be transplanted without even immunosuppression. And the recipient lived for 8 years, and the donor had lived until 2011 and he died at the age of 79. So they proved that even it's not, doesn't have a huge impact on the donor as well as successful in the recipient. But then they thought of it's time now to study the immunology and understand how this immunosuppression works. So I'm just going to switch gear now and talk a little bit about the field of immunology. Of course, I'm not going to go through the definition, but I would like to give an idea about the British physician whose name is Edward Jenner, who had played a major impact on development of the field of immunology. And if you open any immunology book, you will find his picture. And under his picture, they put the title of Father of Immunology in the 18th century. So what did the Edward Jenner discover? So in the Jenner time, there was a virus called smallpox. This smallpox killed around 10% of the population, with the number exceeding up to 20% in some towns and cities. And infections were very easily spread because once the vesicles rupture, it can easily infect any other person come in contact with the disease one. But at the same time, Edward Jenner noticed that there is another similar disease which infects the cows. And this disease called cowpox, it causes also small vesicles in the cow. And humans who deal with the cows, like milking the cows, also get infected by this cowpox, but it's not a fatal disease. And at the same time, he also discovered that patients who got infected with cowpox, they never get smallpox, or even if they got smallpox, it's not fatal for them. And also, this is just like a picture of the Egyptian mummy died from smallpox. So smallpox is not a recent disease. It's a very ancient disease and a lot of stories behind it. So this is actually the experiment that Edward Jenner did in his city. So you can see in his home or his clinic, there are two maids. The one behind him was a maid who was working in the farm, milking cows. And you can see he took a blister from her hand, and she's covering her hand with some sort of napkin. And the maid in front of him is the mother of the child that you can see. And he injected one of the smallpox from the cowpox into the young child. And in the next day, he injected the child with the smallpox in the other hand. And surprisingly, the child survived and was not infected by smallpox. Of course, this cannot happen these days. We have more ethical stories, but if he didn't do this experiment, we will not move in our field. And during this time, at Edward Jenner, they advertised in all the cities that all people must get vaccinated by the cowpox vaccine, and they were like going and advertising, and if you didn't get the vaccine, you would be kicked out of the city. So as normal, there was anti-vaxxer, and people were get frightened of this vaccine, that they were even thinking if they got the vaccine, they will see some of the cow coming out of their hands, or their nose is going to be transformed into a cow, or even coming from their butt here, or from their tongue. But actually, this picture proves that vaccines work. And this picture of two friends, one of them got the vaccine, and one of them didn't get the vaccine, and vaccine works. Although Edward Jenner is one of the modern or kind of modern scientists, Thucydides described the plague of Athene in 430 BC. And this is his description in the Old English. Let me read it for you. So, yet it was those who had recovered from the disease that the sick and dying found most compassion. These knew what it was from experience and how no fear for themselves. For the same man was never attacked twice, never at least fatally. So the history of immunization or infection was known even 430 BC. So let's move now to the modern practice and explain how the field of immunogenetics has been transformed. So this picture is for Dr. Clarence Cook Little, and he is born in Boston, USA. He was a genius scientist. He actually published 15 papers in science when he was 21 years old, and he was the president of two universities at the age of 40, and he resigned and he founded the Jackson Laboratory, which is instrumental in advancing transplantation. And this genius scientist has revealed the genetic basis of transplant rejection, leading the discovery of the major histocompatibility complex. And for you who never visited the Jackson Laboratory, this is the location of the lab. It's in Maine, USA. It's a very nice location, very nice equipped lab. And it provides mice and embryo mice to the whole world. But let me share with you a story which happened in 1947. So the whole lab got fired. Sorry, like the fire destroyed the whole building, and killed the entire inbred mouse. So this was almost 18 years of work. But Dr. Clearance at this time, which shows a very good leadership, he didn't give up, and he met with his staff, and by his communication, he was able to communicate with all the labs around the world who has used their mice to give them a pair of their inbred mouse. And in 1 year, he built this Jackson lab again, and it became back to normal. So let me share with you some of the early experiment that Dr. Clearance Little has done here. So in 1922, so almost 100 years ago, he was working on a specific kind of mice, which has they have a certain tumor. So he started to take some part of this tumor tissue and transplanted this into a healthy mouse of the same strain. And he found that this tumor survived. But when he repeated the same experiment using a different strain, this mice rejected the tumor. So from his first experiment, he said, okay, the recipient must provide factors that match those required by the donor tissue. And the conclusion from his second experiment, he said, loss of tissue transplant might be due to the expression of something harmful by the donor rather than by absence of something beneficial in the recipient. So in 1941, is another scientist who won another Nobel Prize here, Dr. George Snell, and he was an American scientist. He joined the staff of the Jackson lab in 1935, and the reason they hired Dr. Snell was to continue the work that Dr. Snell has started. But Dr. George Snell was more interested in the field of transplantation rather than the field of tumor. So he repeated the same or very similar experiment that was started, but instead of using tumor, he transplanted a skin into the same strain of mice, and the skin graft was accepted. But when he transplanted a different strain of mice into a strain of a different strain into a different strain, the rejection happened. So Snell was studying graft resistant genes, and he called this histocompatibility, or the H gene, at this time. So he repeated the same experiment again, but make it more challengeable. So he made two different strains, strain A and strain B, and then the offspring now of this mated was A, B, and AB, and he took a skin graft from the parent into the offspring, and he found that the graft was accepted in the offspring, but when he went backwards from the offspring back to the parents, the graft were rejected. Of course, it was rejected because you can see here the offspring see the A gene in the family as known, but the parent doesn't see the B as a matched. Sorry, the parents see the B as a mismatched antigen, and this is why rejection happens. And in a different part of the world was Dr. Gorer. He was a British immunologist. He won also the Nobel Prize in 1960. And one time he was very bored in his lab, and he said, okay, let's see if the mice of the red blood cells of the mice also express different antigen as in humans. So what he did, he bleeded himself, and he isolated the serum from his blood, and he started to get 3 different strains of mice and mixed his own serum with the different strains of the red blood cells. And what he found, that the strain CBA act very weakly. Strain A agglutinated, so his serum agglutinated the red blood cells, and strain C57BL did not agglutinate, the serum didn't agglutinate the red blood cells. So he concluded that also mice can present with different blood groups, same as human. But in order to bleed himself continuously, this was a lot of work, and he found an easier situation for that. So he immunized rabbits with the erythrocytes from these mice, and then he sacrificed these rabbits and then collected the serum from these 3 different mice. So the mice who was sensitized by CBA become the anti-sera 1. The mice sensitized by the strain A become the anti-sera 2, and the mice sensitized by the 57BL become the anti-sera 3. And he repeated his experiment using these 3 different sera. So anti-sera 1, it didn't have any, sorry, it agglutinates CBA, agglutinates strain A red blood cells, and was weak on the C57BL. Anti-sera 3 actually agglutinated the whole red blood cells. And interesting here, the anti-sera 2 behaved in a very similar way to his own blood. So he concluded that the rabbit anti-serum recognizing antigen 2 behaves similar to his own serum. Mice strain A carry antigen 2. Mice strain 57BL lack of antigen 2. And all his experiment now was carried on these 2 mice, which is mice strain A and mice strain C57BL, which I'm going to present in the next slide. So he used the strain A because this strain A is known to have the antigen 2. And this strain A has a sarcoma that arises spontaneously in this strain. So he took some cells of this sarcoma and he transplanted them into into the same strain A and into the different strain C57BL. And as expected, the sarcoma cell grew in strain A, but it was rejected in the C57BL. So during his staff meeting, there was a lot of negotiation that the rejection was because not because of the antigen 2, but it was because it's just a different strain. So in order to prove his theory, he mated these 2 strains together, the strain A and the strain C57BL, and then he looked at the offspring of these 2 mating. So they will carry half of the genes from the A and half of the genes from the C57. So by Mendelian law, you will have 2 or 50% of the offspring will carry antigen 2 and 50% will not carry antigen 2. So next he took the parent, which is strain A, and also again transplanted this offspring with the sarcoma cells. And he found that only 50% of the mice had the tumor growth and 50% rejected the tumor. So he concluded that antigen 2 shared between the malignant and normal cells apparently was an important resistant factor to the growth of the allogenic tumor when present in the donor and absent in the recipient. So, so now we have 2 Nobel Prize winners. We have the British scientists and we have the American Dr. Snell scientist. So Dr. Little was amazed by the work of the British scientists. So he actually invited Dr. Gurer to come in the States for a 1-year sabbatical. And actually during this 1 year, he joined the Jackson lab. He worked in close with Dr. George Snell, and they came up with the name of H2 resistant gene, which I explained at the beginning of the slide, and then this became the name of the measure histocompatibility in mouse. So basically what they did is they repeated the early experiment that Dr. Little has started, and then they sacrificed the mice and then they took the bloods from this mice, and then they tested the anti-SERA2 on the blood of these mice, and they found that the anti-SERA2 agglutinated the mice where the tumor survived, and the anti-SERA2 did not agglutinate the blood that when the tumor was rejected. So the presence of antigen 2 on the erythrocyte of the host was strongly associated with the growth of the tumor from the strain, and they both mutually agreed to call this resistant gene as the H2. So let's switch gear now again, and let's talk about the discovery of the major histocompatibility in human. And the molecule of the HLA was described in the early 1950s independently by three groups of scientists. The first scientist, the one you see in the picture, is Dr. John Doucet. And he was a French scientist, and he also won the Nobel Prize in 1980. This time, this French scientist noticed that mixing patient serum who were multiple transfused, so this serum comes from a sensitized individual. When they mix their serum with another individual, blood cells, white blood cells, they agglutinate these blood cells. He also noticed that patient serum agglutinates some of the leukocyte and some not. And at the same time, he noticed that it never agglutinates the patient's own white blood cells. And this is just a picture of how an agglutination looks like. So he hypothesized that agglutination is due to the presence of alloantibody that was stimulated by the transfused blood. He liked the idea and started playing with the sera from other transfused patients, and he was able to identify at this time 4 different sera. These 4 sera identify the same antigen on the cells. And he named this antigen as MAC or MAC. And MAC stands for the first letter of the 3 of his volunteers that he started the experiment with. Later on, MAC became known as the HLA. So Doucet later started to call this system HU1, and H also stands for histocompatibility at this time, and 1 because to differentiate it between the mice and human. And he put the letter U because it was the letter from his first name. This is a chart, a working chart from the Doucet lab, and people who used to visit Dr. Doucet lab in St. Louis Hospital have found this chart is all over the wall in his office and his lab. Basically, it's an association studies where every dot here represents 1 cell, and the gray or the light gray is representative when the serum agglutinate the white blood cells, and the dark dots or the dark big dots represent when the serum does not agglutinate the white blood cells. Currently, this method is known, or we'll use it in the HLA lab as what we call the panel reactive antibody. And we use it in the HLA lab to predict the percentage of a patient to react positive to the donor in the population. And the circle here that you see is the clusters of antibody recognizing the MAC, which is now known as the HLA-A2. And HLA-A2 is one of the most common antigen in the Caucasian, and almost 50% of the French population carry these genes. In addition, in collaboration with the transplant surgeon, Dr. Doucet examined the effect of the HU1 antigen injection on skin graft and further confirmed the conclusion that this HU1 complex was indeed a transplantation antigen. The second scientist also who worked on the field of HLA is John Van Rood, and this was during his PhD. And what this young scientist found, he was from Holland. He found that he was also using serum from transfused leukocyte, but he used a simple IBM computer program, which was owned by the Dutch Bureau of National Statistics at this time, to help him to identify the alloantibodies. And computers was a little bit scary at this time. So what you see here in the top right, using the computer algorithm, he was able to cross-react two different serum with large panel of leukocytes. So again, the dark dots represent a positive reaction, and the light dot represent a negative reaction. But using this serum, he was able to identify two main categories, which is and he later named them the 4A and the 4B. And every individual can be either a homozygous for 4A or a homozygous for 4B or heterozygous 4A and 4B. And later on the field, and for people who have seen our HLA report, we use these to identify the antigen. And now it's called the BW4 and the BW6. So it's very interesting to see a full work of a PhD thesis become translated into two just numbers. The third scientist is Dr. Rose Pine in 1963, and this is the paper that she published, which actually laid the foundation of the HLA that we are currently using. So on the left side here, you can see Rose Pine with her group taking advantage of a Van Rook computer system. And they did further development of this system using a computer that was present at Stanford. And on the left side, you can see that this is the 2x2 association table that they did, or the chi-square analysis. And they were able to identify two additional antigen. They didn't know that this antigen is the HLA, but actually they call this antigen the LA1 and the LA2, which is in the red circles. Later, they call them locus antigen 1 and locus antigen 2. So in conclusion, Doucet and Payne were using two different sera because Payne was using sera from pregnant women. So because pregnant woman has been sensitized from the infant, and at the same time, Doucet was using sera from people who has been repeated transfusion, so it's also sensitized. And both of them were using two different techniques, but the end result were the same. One of them called it HU1 and the other one called it LA1 and LA2. So because the field starting to grow, all these three scientists that you see here in the bottom of the picture, Dr. Doucet, Rose Payne, and Van Rook, they collaborated together, and they advertised to anyone else who's interested in this field, and they made the first international histocompatibility workshop, which was done in 1964. And until current days, this is done almost on a yearly basis. And this workshop was a wet bench workshop, and a lot of scientists from different kinds of the world were joining this workshop. Everyone was bringing his own sera and the cells, but the idea here that they give every of these lab 6 sera and 6 blood donors, and they ask them to use their own method because they wanted to speak the same language. They want to standardize the method. So they give them the same sera, the same donor, and ask them to test them using their own method to see which is the best method that will work. They also used functional assays like the mixed lymphocyte reaction. And at this time, they didn't know that there are two classes of HLA, like the class 1 and the class 2. But now we know that the class 1 are detected by serology, and the class 2 are detected by the functional assays. And in the second international workshop, it was in 1965. And the main question here is that they need to speak the same language. So in 1965 and during the second workshop, they decided that we need a committee that can provide us with guidance on the nomenclature. And after the third workshop in 1967, they came up with a new name of this histocompatibility antigen. They call it the human locus A antigen, and human come from H1 from Doucet and LA from Penn. That's why in the beginning of the slide, anyone who were attempt to answer HLA as human locus antigen or human locus A is considered a correct answer as well. And then they published this report in 1968. So because every lab was working on the same field, but every lab was giving it a different name. So this was just like a figure for different labs using different names. So you can see here, Pan was using LA1, LA2, which is the same as the MAC that Doucet was using. But when with the new nomenclature, they give it the HLA-A1 and the HLA-A2. And at this time, they only knew about 8 HLA antigen at this time only. And in 1968, the WHO took over the HLA nomenclature committee, and this committee still exists until these days. They changed the name of to become HLA instead of human locus antigen. So now it's become histocompatibility human leukocyte antigen, because it was clear there was more than one genetic locus are responsible. And they divided the HLA into HLA class 1 and HLA class 2. And this is the latest update, or the update in the end of October 2021 from the WHO Nomenclature Committee. And what you can see here on the x-axis is the number of alleles that has been discovered over the years, starting from 1987 until 2021. And you can see there is the green here is the HLA1 is 23,000 alleles, and the dark green or the dark bars here, the HLA2 is almost 8,000. So a total of 30,000 new allele has been discovered until end of October 2021, and the field is still growing. And I just found it's funny because out of this 30,000 allele, I just found 6 of them. And this was only done in 1 year during when I was validating the next generation sequencing when I was in Calgary. And you can see that the numbers that I received here was a little bit a big number, but and the paper was published in Human Immunology. And this is just like a snapshot of the letter that you receive when you submit a new allele, and then the WHO nomenclature give you the name. So, for example, here this was an allele that we found, and it's called the DQ beta 105 144. And then they ask you if they appreciate if we can send them a copy of the paper once it's get published. So now I'm going to switch gear into more interesting subject and to show you what Saskatoon has added to the field of the kidney transplant. So before 1960, in order to treat patient with kidney failure was only 2 treatments, either giving morphia to the patient and the family, you just give them Kleenex to wipe their tears. But after 1960, and this picture is taken from St. Paul Hospital, was the administration of the hemodialysis unit. But before going into details about this hemodialysis unit, I would like to remind you of the Baltzan family here. So as you see in the picture, this is Dr. David Baltzan, and he was the first specialist of internal medicine cardiology in the province in 1928. And this is the picture of the 3 of his sons. This is Dr. Mark Baltzan. He is a transplant nephrologist. And this is Dr. Richard Baltzan is a nephrologist, and Dr. Donald Baltzan is a surgeon. And now this, they were all graduated from McGill, but I'm going to focus now on Dr. Mark Baltzan. and he received his training after he graduated from McGill in Johns Hopkins Hospital, where he learned the field of nephrology there. So going back to the hemodialysis unit, so during the Second World War, both soldier and civilian experienced acute renal failure following the bombing raid, which later became the crushed kidney syndrome. And the solution was an artificial kidney or hemodialysis machine that kept the patient alive for several days by cleansing the blood until the kidney recovered. And in 1943, Dr. Clough from Germany invented this hemodialysis unit. So when Mark returned to Canada, he convinced his dad to buy the first Clough dialysis machine in Saskatchewan. But before the new equipment arrived to St. Paul Hospital, Dr. Mark received a phone call from Dr. Lou Horlick at RUH asking him with the help with a patient named Alma Kitwhite, which is the one in the picture here. And what happened to this young lady is she got acute renal failure after her delivery with very high potassium count. So because Dr. Mark was very well known, he convinced the military to bring them a dialysis machine, which was in Calgary at this time. And he actually installed the dialysis machine himself in the surgeon changing room in St. Paul Hospital, and he connected the tubes to the shower so that it can flush through the shower. And it was the first time for all of them to experience it. They were injecting, they were using a big 17 gauge needle inserted into the femoral artery, and sometimes it get blocked, and on their own word, they can see splashes of blood all over the wall and all over the place. So it was a very bit messy. But the good news that Alma here got recovered, and she recovered from her acute renal shutdown. So as I mentioned to you, Dr. Mark Paulson, he performed the first dialysis in Saskatchewan in 1960. He also performed the third kidney transplant in Canada in 1963. And this was the actual challenge. And this is a picture of the 19-year-old Stella Mossing. Her kidney stopped function. And Stella came to the Balson Clinic in November 1963 because she had heard about their dialysis machine, and he hoped it might save her life. The Balson dialyzed her, but it was a stopgap measure. At that time, such treatment was never administered to patients whose kidney were diseased and beyond repair. It was only for acute renal failure. So this case frustrated Dr. Mark Balson, and he started to think about the kidney transplant because he heard about it during his training in Johns Hopkins. And he said whether it's possible to be a solution or treatment for Stella Mossing. So Dr. B.T. Lowe was Dr. Balson's wife at this time, and she did a literature review, and she found that there was only 15 kidney transplant in the worldwide at this time. And also she found that there is some drugs that can suppress the transplant rejection. But so this become, Dr. Mark was very convinced, and he started to form a committee of ethics, which include lawyers, people from public, religious people, because in order to take a kidney from a diseased person to put him in a live person, this was a bit of, need a little bit of ethics approval. And in 1 month, they were all convinced that they can go with the surgery. And actually the surgery happened on December 10, 1963, so shortly before Christmas. And Dr. Mark Balson was inside the theater watching it. And as soon as they anastomosed the artery and the vein, the kidney started to work. And Dr. Mark was very relieved. He went back to sleep. But unfortunately, 12 hours later, they called him and the kidney was rejected. And at this time, it was because of the acute rejection. So The only solution now was to keep Stella alive during the Christmas and to pass this period. And in early 1964, the only solution for Stella was to give her another transplant from deceased donor. But at this time, they gave her an immunosuppressive drug, which is called Immuprone at this time. And they didn't announce to the media until 1 month. But the poor patient died 4 months again from the side effects of the immunosuppressive drug. And so now to show an appreciation, this is the HLA lab, which is now in St. Paul Hospital, and it's actually started as one bench or a small bench in transfusion medicine at RUH. Now we have our own lab, and you can see we have the picture of Dr. Mark Balson on the lab because the family was initially who founded this lab. And if you open the door, you will find the lovely team waiting for your service. From left to right is Carrie, Destiny is the lab supervisor, Twyla, Tech 2, and our manager, Giselle Skoski. Carla was a temporary, now she is back to hematology, and we have Kathy and myself. And our lab is an ASHI accredited lab, and we are one of the 14 labs in Canada who is accredited by ASHI. We do in the lab here risk assessment for renal transplant and bone marrow transplant, either pre or post. We also do pharmacogenomics and disease association studies. And I would like to share with you some of the pictures from our lab in 2021. We have an automated DNA extraction. We also have the high-end technology for HLA typing using the sequence-based by NGS. We are also having the fastest technology for HLA typing. You can get your typing done in 90 minutes. And we also do molecular HLA typing using the SSO by Luminix. We do also antibody screening by the Luminix 200, and the flow cell cross-match, we have our own using the Cytoflex. We also have a biosafety cabinet and other equipment in the lab. So I would like to thank you for your patience and your time, and share with you the last history of the COVID-19 100 BC. I would like to answer all your questions now.
Thank you, Ahmed. Anybody has questions, comments?
Victor, I'd like to make a comment, though.
Okay. Go ahead, Jay.
Excellent talk from a history point of view, and I'm especially impressed with Mark Balson's story you shared with us, because I've heard it from his own mouth, and I was involved with whenever he was talking about creatine clearance and other things. I'm a medical biochemist, so we used to have a very healthy discussion at coffee break. On those days, we used to have a coffee break around 10:15 or so up at the doctor's lounge. So I'm very highly impressed with your capturing this in a very pictorial way, because you have taken it from step by step, and it's a good knowledge. Nowadays, we don't hear about it, history, and we don't hear about the development. So I want to congratulate you for this excellent talk, especially in Saskatchewan and Saskatoon involvement on a world stage. Thanks very much.
Yeah, something I just forgot to add that actually by the end of 1960, Saskatchewan was doing 10% of the transplant worldwide. This is something, it's amazing actually to hear about as well.
Yeah, because there is another gentleman who was just inducted in 3 years ago in the Medical Hall of Fame, in Canadian Medical Hall of Fame. His name is gone out of my mind at the moment. He was also involved in the kidney transplant up here with Mark Balson and others. So great.
Well, I see several people also echo Jay's sentiment. Fergal, Ahmed, superb review, and then from Mary. Thank you, Ahmed. It is so important to know about our history and the progress of where we are going. And from Tama, that was extremely interesting. Thank you for this fantastic presentation. So anybody else wants to add or ask question?
Sometimes when you don't have questions on it, it's because the work is too over our head to even formulate a proper question to ask. I am curious how anybody even is able to do any work in HLA if they can't bleed themselves and use their own blood to do the experiments on anymore. It must be very difficult to get ethics.
And actually, I would like to take this opportunity and invite anyone who hasn't visited the HLA lab so far to come and join, come for a cup of coffee, show you some of our new methods that we are using, and we are still evolving more and more now.
Well, thanks, Ahmed. It was really kind of fascinating presentation and tour through the history. By the way, the person who actually coined the term transplantology, it was the, I think, Russian guy, his name is Vladimir Demihov, was never recognized. So never got any Nobel Prize or any other prize except a small reception, I think in 80s when he was old. So and hopefully everybody else in this field gonna be in much better situation. So again, thank you, Ahmed. And with that, we're gonna conclude our grand rounds for this year. There's gonna be a break for December, and then we're gonna start in January our rounds. And I would encourage everybody to participate in that, and also send your feedback. So shortly we're gonna issue CME credits, and for those who participated in Dr. Omar Shakir lecture, Fergal told me that we can issue also credits.